If every single person on Earth vanished tonight, except for one small group standing on the red dust of Mars, how many of them would need to survive for the human race to still be here in 10,000 years? Not to just survive the next winter, not to grow enough food for a few decades, but to still be a healthy, walking, thinking, laughing species 12,000 years from now, with grandchildren who don't carry the same genetic mutation, with immune systems strong enough to fight off a disease nobody on that planet has ever seen, with enough different eye colors and blood types and bone structures that a geneticist looking at their DNA 1,000 years later would never be able to tell they all descended from a tiny, desperate colony. Most people guess a number in the thousands. Some guess in the millions, because that is what science fiction has trained us to imagine: giant generation ships packed with tens of thousands of colonists drifting between stars. But the real answer, the number that comes out of population genetics, medical statistics, and decades of actual research into minimum viable populations, is so much smaller and so much stranger than anything Hollywood has ever shown you. And once you understand why that number is what it is, you will never look at the phrase, starting over the same way again. This is not a thought experiment dressed up as science. This is a question that space agencies, geneticists, and mission planners are actually working on right now. Because if humanity is serious about becoming a multiplanetary species, this number is not a philosophical curiosity. It is an engineering requirement, as real and as unforgiving as the amount of oxygen a habitat needs to hold, or the amount of shielding required to block cosmic radiation. Get this number wrong, and you don't get a failed business venture or a delayed timeline. You get a colony that looks successful for 2 generations, and then quietly, invisibly, begins to collapse from the inside, not because of a meteor strike or a life support failure, but because of something written into every strand of DNA in every person who ever stepped off that ship. So let's actually do the math. Let's walk through where this number comes from, why the old assumptions about it were dangerously wrong, what has to be true about the group of people who go, and what it would actually take, biologically, socially, and logistically, to plant a seed of humanity somewhere else in the universe that has a real chance of growing into a forest. We need to start with a concept that most people have never heard of outside a genetics classroom, called the minimum viable population, or MVP. This idea did not originate with space travel at all. It came from conservation biology, from scientists trying to figure out how many California condors, how many black-footed ferrets, how many mountain gorillas needed to survive in the wild for that species to have a good chance of avoiding extinction over the long term. The logic is brutally simple once you see it laid out. A species is not just a collection of individuals. It is a pool of genetic variation. Every individual carries a slightly different combination of genes, some helpful, some neutral, some silently harmful but masked by a healthy copy from the other parent. When a population is large, that pool is deep and wide, and harmful mutations tend to stay rare and hidden. But when a population shrinks, especially when it shrinks suddenly, something called a genetic bottleneck happens. The gene pool that survives that bottleneck is a random, incomplete sample of everything that existed before. Some good genes are lost forever, by pure chance. Some bad genes, by pure chance, become common, because in a tiny founding group, even a single person carrying a harmful recessive gene can eventually pass it to a large fraction of all future descendants. This is not theoretical. We have watched it happen in real animal populations. Cheetahs today are famous among geneticists for having almost no genetic diversity at all, the result of a massive population crash roughly 10,000 years ago. Cheetahs from opposite ends of the African continent are more genetically similar to each other than siblings are in most other species. The consequence is a population with weakened immune systems, high rates of sperm abnormalities, and a species that is far more vulnerable to disease outbreaks than it should be, because there is no genetic backup plan. If one cheetah is susceptible to a particular virus, they almost all are because they are, genetically speaking, almost the same individual repeated over and over. That is what a bottleneck does. It doesn't just shrink numbers, it shrinks the future. Now bring that concept to a group of human colonists standing on Martian soil, or on a terraformed moon, or inside a rotating space station orbiting a star that is not our sun. That founding group is, by definition, a bottleneck. It is a random, tiny sample of the entire genetic diversity of Earth's 8 billion people. The question conservation biologists, and more recently, anthropologists and mission planners have tried to answer is, how large does that sample need to be so that the resulting population doesn't slide into the same trap as the cheetahs? So that 100 generations later, their descendants are not all carrying the same 3 or 4 fragile genetic weaknesses, with no diversity left to fight off whatever new pathogen or environmental stress comes their way. For decades, the rough rule of thumb used in conservation biology was something called the 50-500 rule. 50 breeding individuals to avoid the immediate, short-term dangers of inbreeding, and 500 to maintain enough genetic diversity to keep evolving and adapting over the long term. That rule has actually been revised upward by more recent research. Some scientists now arguing that the long-term number should be closer to several thousand to truly guarantee evolutionary resilience over centuries. But here's the twist that changes everything when you apply this to human space colony, rather than a wild population of condors or ferrets. Wild populations rely purely on random mating and natural selection to manage their gene pool. Human colonists do not have to. Human colonists can plan. This is where the research on human minimum viable populations diverges sharply from the raw biological numbers, and where things get genuinely fascinating. In the mid-2000s, anthropologist John Moore, working from a background studying isolated human populations and the history of small founding groups throughout human migration, proposed a strikingly small number for a viable interstellar or interplanetary colony, somewhere in the range of 160 people. Later refinements, run through detailed computer simulations by researchers including Cameron Smith at Portland State University, modeled generations of a colony forward through time using known human demographic patterns, fertility rates, mortality rates, and genetic inheritance, and came out with figures generally in the range of about 98 to a few hundred individuals as a workable lower bound, provided that certain conditions were met. Not just any 98 people, a carefully selected, carefully managed 98 people. That phrase, carefully managed, is the single most important idea in this entire subject, and it is the part almost every popular article about this topic completely leaves out. A minimum viable population is not simply a headcount. It is a headcount combined with a set of rules about who reproduces with whom, over how many generations, and how strictly those rules are followed. Simulations like Smith's don't just ask how many bodies you need. They model actual mating strategies across 100, 200, sometimes as many as 10 generations, and they test what happens to genetic diversity and inbreeding coefficients under different reproductive strategies. A colony of 160 people who pair off randomly and reproduce however they choose, generation after generation, will almost certainly not remain genetically healthy for very long, because random pairing in a tiny population inevitably means cousins marrying cousins within just a few generations, since there simply aren't enough unrelated partners to go around. But a colony of the same size, following a managed breeding structure, perhaps built around careful record-keeping of ancestry, perhaps supplemented by frozen genetic material, perhaps guided by simple rules limiting how closely related any two parents can maintain healthy diversity for far longer than random chance would ever allow. This is where the conversation moves from pure genetics into something almost uncomfortable to talk about, but essential if we are being honest about what this actually requires. Mission planners and researchers who study this seriously have proposed ideas that sound cold on paper, but are really just applied statistics. Things like ensuring the founding population has a roughly even ratio of men and women of reproductive age. Things like limiting the number of children any two closely related family lines are allowed to produce together relative to the total population, simply to prevent any one genetic lineage from dominating the future gene pool too heavily. Things like bringing along a deep bank of frozen sperm and eggs, and in more speculative proposals, even frozen embryos, sourced from thousands of donors back on Earth, specifically to inject additional genetic diversity into the colony at intervals over the following decades and centuries, long after the original ship has landed and its supplies have been consumed. This is not colonists deciding who falls in love with whom. Love, relationships, family structure, all of that remains entirely a matter of personal choice, exactly as it should be. What we're describing here is a supplementary system, a genetic insurance policy running quietly in the background, similar in spirit to a seed bank, except instead of protecting crop diversity, it protects human diversity, giving mission planners a tool to correct for genetic drift without ever dictating anyone's personal life. Let's talk about why the diversity itself actually matters in practical medical terms, because this is not an abstract concern about ethnicity or appearance. Genetic diversity within a population is directly tied to immune system strength. Humans carry a set of genes called the major histocompatibility complex, or MHC, which is responsible for how our immune system recognizes and fights off pathogens. Populations with high MHC diversity are able to recognize a much wider range of viruses and bacteria, because different individuals carry different immune recognition profiles, meaning a pathogen that slips past one person's defenses might be caught immediately by another's. A population with low MHC diversity is, in a very real sense, an army with only one uniform and one set of tactics, and any enemy that figures out how to beat those tactics can beat everyone. On a new world, exposed to genuinely unknown biological and environmental stresses, whether local microorganisms, altered radiation exposure, or diseases that mutate differently under different gravity and atmospheric conditions, this kind of immune diversity is not a luxury. It could be the single deciding factor in whether an outbreak becomes a manageable illness or an extinction-level event for a colony with nowhere else to turn for help. Beyond genetics, there is the second half of this equation that most people never even think to ask about, which is the question of skills. A colony cannot survive on genetic diversity alone, because genetic diversity does not grow food, does not fix a broken oxygen recycler, does not deliver a breech birth safely, does not teach the next generation how to read or how to think critically or how to resolve a dispute without violence. If you were designing this founding population from scratch, purely for genetic health, you might end up with a wonderfully diverse but practically useless group of people, none of whom know how to weld, how to farm in artificial soil, how to perform surgery, or how to repair a solar array in negative 80-degree cold. So the real number, the one mission planners actually work with, is a delicate overlay of two separate requirements stacked on top of each other. The genetic floor, which research suggests sits somewhere between roughly 100 and a few hundred individuals under a well-managed system, and the functional floor, the minimum set of skills and professions required to keep a self-sustaining settlement alive and growing. Think about everything a fully self-sufficient settlement needs, completely divorced from Earth's supply chains. It needs people who understand large-scale food production under artificial or partially artificial conditions, agronomists and horticulturalists who know how to grow calorie-dense, nutritionally complete crops in greenhouses or hydroponic systems, because a Martian colony cannot simply plant a field of wheat outside and wait for rain. It needs structural and mechanical engineers who understand how to build and maintain pressurized habitats capable of withstanding a hostile outside atmosphere indefinitely. Because a single major structural failure in an airtight seal is not an inconvenience, It is a mass casualty event. It needs medical doctors spanning general practice, surgery, obstetrics, and psychiatry, because childbirth, injury, and mental health crises do not pause themselves while waiting for a supply ship that might take years to arrive from Earth, if it arrives at all. It needs power and energy specialists who understand nuclear, solar, or other generation systems well enough to keep the lights, heaters, and life support running through the planet's harshest seasons. It needs chemists and materials scientists capable of manufacturing tools, spare parts, and eventually more complex machinery from local raw materials. Because you cannot ship an infinite supply of replacement parts across tens of millions of miles. It needs educators, capable of raising children who understand not just survival skills, but the deep accumulated knowledge of human civilization, mathematics, history, ethics. So the second and third generations are not simply survivors, but genuine inheritors of everything humanity has learned. And it needs, perhaps most overlooked of all, people trained in conflict resolution, psychology, and governance. Because history has shown again and again that isolated communities under extreme stress do not fail first because of technology. They fail first because of people. That last point deserves its own moment, because it is the part of this story that pure genetics and pure engineering completely miss. And it comes from a real historical case that every serious researcher on this topic eventually references. In 1789, following the mutiny on the HMS Bounty, a small group of mutineers and Tahitian men and women settled on the remote, previously uninhabited Pitcairn Island in the Pacific. The founding population was tiny, only about 28 people. Within just a few years, the colony had descended into a horrifying cycle of violence, alcoholism, and murder, reducing the population dramatically before any kind of stability took hold. It was not a genetics problem. 28 people is a small gene pool, but the colony's near collapse in its earliest years had nothing to do with DNA and everything to do with the absence of functioning social structure, leadership, and conflict resolution among a small group of deeply stressed people, isolated from any outside authority or support. The population that eventually stabilized and grew into today's Pitcairn Islanders survived not because their genes were perfect, but because, eventually, structure and cooperation won out over chaos. This is the exact same lesson mission planners studying human space settlement keep coming back to. You can solve the genetics. You can solve the engineering. If you do not also solve the social architecture, the governance, the psychological support systems, the shared sense of purpose that holds a tiny, isolated community together through the darkest, most claustrophobic years of its existence, none of the rest of it matters. A colony that is genetically perfect but socially fractured will still tear itself apart. So when you put all of these layers together, the genetic floor, the skills floor, and the social floor, you start to understand why serious estimates for a robust, self-sufficient colony tend to land not at the bare minimum of 98 or 160 people, but considerably higher, often cited in the range of several hundred to as many as 10,000, depending on how much risk you are willing to accept and how much outside support. In the form of supply missions or genetic material from Earth, you assume will continue over time. The higher end of that range essentially removes the tightest bottleneck risk almost entirely and provides enough specialized workers that the colony is not catastrophically vulnerable if a handful of key specialists die in an accident. The lower end represents a genuine, calculated gamble, a population small enough to be transportable with current or near-future spaceflight technology, but large enough, if perfectly managed, to have a real statistical chance of long-term survival. This tension between what is biologically ideal and what is logistically possible is really the whole story of space colonization in miniature. Launching humans and cargo off of Earth is staggeringly expensive and staggeringly difficult. even with the most optimistic projections for reusable rockets and in-orbit refueling. A colony of 10,000 people, with all of their food, water, equipment, and infrastructure, represents a logistical undertaking on a scale humanity has never before attempted, likely requiring decades of preparatory missions before a single permanent colonist ever sets foot on the surface. A colony of 160, while still an enormous undertaking, is at least within the outer edge of what serious mission architectures, like those proposed for early Mars settlement, could plausibly deliver within a single human generation of effort. This is why the smaller numbers, the 98 to 200 range, keep appearing again and again in serious space colonization research. Not because scientists believe smaller is safer. They know smaller is riskier. But smaller might be achievable, and a risky plan you can actually execute will always beat a perfect plan you can never afford to launch. There's another layer to this that rarely makes it into casual conversations about Mars colonies, and that is time. Genetic health is not a snapshot. It is a trajectory. A colony of 160 people might look perfectly healthy in its first generation. The real test comes 2, 3, 5 generations later, when the initial diversity brought from Earth has either been carefully preserved through managed reproduction, or has already begun eroding through the unavoidable randomness of who has children with whom, how many children each couple has, and simple bad luck in the form of accidents, illness, or infertility, removing entire genetic lines from the pool. This is why nearly every serious model of colony viability is not a single number, but a range of outcomes, expressed as probabilities. A population of 160 might have, say, an 80% chance of remaining genetically viable for 10 generations under a well-managed breeding strategy. But that same population under completely random pairing might see that probability collapse to something far lower. Scientists modeling these outcomes use tools borrowed directly from population genetic simulations originally built for endangered species conservation, adapted to account for human lifespans, human fertility windows, and human social behavior. And what those models consistently show is that the deciding factor is almost never the starting number alone. It is the quality of the management system layered on top of that number, sustained faithfully across generations, long after the excitement of the initial landing has faded into the routine, exhausting, multi-generational reality of simply keeping a small world alive. It's worth pausing here to appreciate just how strange and important this makes the founding colonists themselves. These would not simply be astronauts or engineers in the traditional sense. Every single person selected for a mission like this would, in effect, be chosen as a living, breathing contributor to the future genetic health of an entire branch of humanity. Their medical history, their family history of hereditary conditions, their genetic screening results would matter just as much as their professional qualifications. Space agencies and private companies seriously exploring long-duration, multi-generational missions have already begun discussing the ethics of exactly this kind of selection process, and it raises genuinely difficult questions that current bioethics frameworks are only beginning to grapple with. Who decides the criteria for genetic screening? What happens if someone develops a hereditary condition after the mission has already launched? How do you balance the cold statistical needs of a colony's long-term genetic health against the deeply personal, deeply human right of any individual to have children with whoever they choose to have children with? There are no fully settled answers to these questions yet, and that in itself tells you something important. We are, right now in this decade, having the opening conversations about a problem humanity has genuinely never faced before at this scale. Let's bring this back down to earth for a moment, quite literally, because there is a natural historical parallel that helps make all of this feel less abstract. Human beings have actually done something very similar to this before, just not deliberately and not on another planet. genetic and archaeological evidence increasingly supports the idea that the initial human populations that migrated out of Africa tens of thousands of years ago and later crossed into the previously uninhabited Americas via the Bering Land Bridge were themselves remarkably small founding groups, in some estimates numbering only in the low thousands, or, according to some studies, of very early migration waves, potentially even smaller. Every single indigenous population across two entire continents, from the Arctic Circle down to the southern tip of South America, descends from that tiny founding pool. It worked. Humanity did not collapse from lack of genetic diversity during those migrations, and those populations went on to build sophisticated societies, agriculture, astronomy, complex governments, all from a starting gene pool that, by modern standards, would look alarmingly small. This is actually one of the most encouraging pieces of evidence available to space colonization researchers, because it demonstrates that human populations have survived exactly this kind of genetic bottleneck before, under far more primitive conditions, with none of the modern genetic screening, medical support, or careful management strategies a planned space colony would have available. If our ancestors could do it by accident, with nothing but luck and resilience, a planned, managed colony equipped with genetic screening, medical technology, and centuries of accumulated scientific knowledge about exactly this problem should, in theory, have a considerably better shot. Of course, should, in theory, is exactly the kind of phrase that separates comfortable science communication from the messy, uncertain, actual work of mission planning, and it's worth being honest about what we still don't know. We don't know, with certainty, how human fertility and reproductive health will function long-term under partial gravity, whether on Mars at roughly 38% of Earth's gravity, or in a rotating space habitat with artificial gravity that may not perfectly replicate Earth conditions. Some research on animal reproduction in microgravity environments has shown concerning effects on fertility and embryonic development, though results remain limited and far from conclusive for humans specifically. We don't know exactly how radiation exposure beyond Earth's protective magnetic field will affect mutation rates across future generations, though we do know that unshielded, long-term radiation exposure is a serious concern that any real mission would need to solve through habitat design long before it became a genetic issue. We don't know exactly how isolation, small population psychology, and the sheer, crushing weight of knowing you and your descendants can genuinely never simply go home again will affect the mental health of a founding generation over years and decades. Every number discussed in this video, whether it's 98, 160, or 10,000, comes from careful modeling, historical parallels, and genuine scientific research. But it also comes with real uncertainty attached, because nobody in the history of our species has ever actually attempted this before. And maybe that is the most honest way to end this conversation. The question of how many humans it would take to restart civilization on another world is not really a single clean number waiting to be discovered, the way you might calculate the exact boiling point of water at a given altitude. It is closer to a design target, a moving goalpost shaped by genetics, medicine, engineering, psychology, ethics, and the raw economics of getting anything at all off of this planet and safely down onto another one. The scientists who study this seriously will tell you, again and again, that smaller founding populations are not impossible, but they are fragile, requiring a level of careful, sustained, multi-generational management that no human society has ever successfully maintained before, precisely because no human society has ever needed to. Larger founding populations are safer in almost every dimension, genetically, socially, functionally, but they push the logistical and financial requirements of the mission itself into territory that remains, for now, far beyond what any space agency or private company can realistically deliver. What we do know with real confidence is this: it is very likely not one person, not a dozen, not even a few dozen. And it is almost certainly not the tens of thousands of colonists that popular science fiction has trained generations of readers and viewers to imagine. Somewhere in that surprisingly narrow, carefully studied range, somewhere between roughly 100 people managed with extraordinary discipline and a few thousand people managed with reasonable care, lies the actual number, the real answer to a question that sounds like science fiction, but is quietly already being worked on by real geneticists, real anthropologists, and real mission architects, preparing for a future that may arrive sooner than most of us expect. The next time you look up at Mars, a small reddish point of light in the night sky, consider this. Somewhere in the research papers being written right now, in university labs and space agency planning documents, someone is trying to calculate the precise, minimum, carefully balanced number of human beings it would take, standing on that distant red world, to make sure our species never truly runs out of chances. And whatever that number turns out to be, it will be one of the most important calculations our species has ever made, because it will not just decide the fate of a mission. It will decide whether humanity, as we know it, has a future beyond the one small, fragile planet we have always called home. If this kind of deep, real science fascinates you as much as it fascinates me, make sure you are subscribed, because we are going to keep exploring the questions science fiction only pretends to answer. And reality is often far stranger and far more hopeful than fiction ever dared to imagine.